therapy trials in humans had achieved relatively little besides notoriety. But that year, Dr. Jean Bennett, Professor of Molecular Ophthalmology at the University of Pennsylvania School of Medicine and a Gold Fellow of ARVO, presented the results of a historic study at the ARVO annual meeting. ARVO was overrun by reporters and news teams as her work made headlines across the world and even in Australia. Jean and her large team of basic scientists and clinical researchers had successfully replaced a healthy copy of the RPE65 gene in eyes of patients with a retinal dystrophy known as Lebers Congenital Amaurosis, and they had restored vision. Today, Jean and members of her team will tell us about their experiences in developing retinal gene therapy. Please join me in welcoming Jean Bennett.
Thank you so much, Justine, and I'd also like to extend my thanks to the trustees of ARVO and the many, many other people who have made this event happen. It's such an honor to be here, and I'm so grateful and so humbled. And it's also an honor to be the first woman to present a keynote address at ARVO, although it's surprising to me that there haven't been others. But hopefully I won't be the last, and at least for now, I'm the best. So I've had such an interesting and fulfilling career, and it's so many gratifying experiences that scientists don't usually have. And it's all because of team effort. And so the only reason why I accepted doing the keynote address is to present things from a team perspective with the different views of the different experiences of the different individuals. In fact, there are far more individuals working on our team than ever will be treated in our clinical trials. And this journey has been through uncharted territory, and there have been many challenges and surprise, but we've kept our ball on translating the research in the laboratory into the clinic. And obviously, this is the ultimate goal-driven research. So today, we'd like to share some of the experiences that hopes that other groups can benefit from our journey and move faster to develop cures. So to reflect on our different perspectives, this presentation will be interactive. And it'll be a team presentations with bench scientists, clinicians, and our sponsor, as well as the real pioneer, who is the first child in the world to enroll in a gene therapy trial for a non-lethal disease. He really is the true pioneer. And so I want you to think of questions as we go along first, and then we'll have time at the end for questions for anybody in the team. So I'm going to lead the way first by presenting the background information, the current status of our studies, and spicing it up a little bit by some of my own personal observations. And then the other people listed on the front slide will follow suit. So we're grateful for support from many, many groups, especially from the Children's Hospital of Philadelphia, and also from the Center for Advanced Retinal and Ophthalmic Therapeutics, known as CARET, a newly established center at Shea Eye Institute, University of Pennsylvania. And I'd also like to disclose that my husband is Albert McGuire, and he is the surgeon on all of our clinical trials, and the PI of the trials. And both of us are co-authors on a patent regarding this work, but we both waived any potential financial gain in 2002. And you can see my other potential conflicts listed at the bottom of the slide. So just to summarize the results we have, I just like, a picture is worth more than 1,000 words. We have seen such amazing results. This shows one of the children enrolled in our gene therapy studies before treatment, in fact, 2 days before his first intervention. And this is the same child a year and a half after the single eye intervention, being able to do things independently, ride his bike to his friends' houses, play sports, read books, and be in a normal classroom. And this is representative of most of the children who have been enrolled in our study. and is what makes us keep going. So, thank you. So, what is this condition? This is called Lebers Congenital Amaurosis. It's a rare, very early onset retinal degenerative disease. It's usually first identified in children, in infants, and they have very, very poor vision, whatever is measurable from early in life. And whatever vision they have degenerates over time. And shown here are views of the eyes of a child with Lebers Congenital Amaurosis due to RPE65 mutations, and also a dog. And you can see there's nystagmus, which reflects the fact that the retina isn't telling the brain much signal, and the brain's then not holding the eyes steady. What is the reason why this disease progresses? Well, this particular form is due to mutations in a gene called RPE65, stands for retinal pigment epithelium 65 kilodalton protein. This is a gene which is expressed in the retinal pigment epithelium, which helps in the vitamin A cycle. It recharges the retinal, so it provides 11 cis-retinal to the photoreceptors, and without that, the photoreceptors can't function. So from a gene therapy perspective, this is really quite simple. The idea is to deliver the normal copy of the RPE65 gene to those retinal pigment epithelium cells and bypass the defect. The treatment strategy is to package the recombinant DNA inside of a virus, a recombinant virus called adeno-associated virus, or AAV. This is a virus which in wild-type form has never been shown to cause any disease in animals or humans. And it's injected subretinally, as shown in the cartoon, delivering the virus so that it reaches high concentration to the cells that need treatment, the retinal pigment epithelium cells. And depending upon how much volume is injected, there's different amounts of the retina are exposed to the virus. And amazingly, this localized retinal detachment flattens down really quickly, so that by the next visit, there's very little evidence that there's been any injection at all. So to put in perspective what we've done, of course, builds on previous science, and we started working on gene transfer to the retina about the same time when the first genes known to cause retinal degeneration were identified, long time ago, 1990. And shown here, in fact, is Al McGuire with Greg Ackland developing the surgical procedure in dogs in a few years in the early 1990s. And the RPE65 gene had not even been identified at that point. In fact, it was as a cause of disease. In fact, it was first identified as a cause of human LCA in about 1997, and soon thereafter the cause in the dog followed. And we and our collaborators injected 3 dogs which have this condition, or born spontaneously with this disease, and they began to see, and that launched the whole study. And our collaborators started a clinical trial, aimed to start a clinical trial in Florida, aiming to treat adults. And then one day in July 2005, I was really amazed when Dr. Catherine High, who's sitting at this table, knocked on my door and said, "Jean, how would you like to run a clinical trial at CHOP?" And I was just flabbergasted, and by the time I got my composure back, I said, of course, yes. What we'd been dreaming about doing is carrying out a clinical trial in a pediatric population, since this is an early onset and degenerative condition, and CHOP is one of the best hospitals in the world for pediatric disease. So we went lickety-split, and in no time, in 2007, we enrolled our first patient and finished the phase 1 clinical trials and began the phase 3 clinical trials more than a year ago. And I'll get to that in a second. So one of the first questions we had was in terms of risk-benefit ratio in terms of age, and that's of course why the pediatric population is so important. Of course, with age, the disease progresses so that the retina looks a lot worse in older individuals than younger individuals, reflecting the loss of cells that would be viable and rescuable. But further, we had a robust data series showing the ability to rescue young animals more efficiently than older animals. And shown in the bottom are videos from dogs that were treated as puppies. And you can see that before the treatment, the dog was stumbling around, and after treatment, there's no problem. One of the problems, though, that we faced is that new drugs are generally not tested in children. And so we found out that we had a lot of work to do in terms of developing a path for pediatric gene therapy and drug development. We had no idea what we were in for. We had to learn a lot about the legislature and drug development principles and assess the risks for children in gene transfer and learn a lot about the laws spelled out in the Code of Federal Regulations. But we worked through that, and then when we presented our proposal to the Recombinant DNA Advisory Committee, the so-called RAC, in December of 2005, we were very fortunate to, after our presentation, to have a couple in the audience who are parents of a child with LCA. Just to give you a little view of the drama there, at the end of this presentation, an ethicist got up and said, well, it sounds all really good, but I just want to let you know that my uncle is blind, and he learned how to navigate the subways of New York City, and he plays a saxophone, and he's a millionaire. It's okay to be blind, which is not what we wanted to hear after our presentation. But fortunately, the Brints got up and gave such an articulate and meaningful speech describing why we need to develop treatments for blindness, how many people it takes to get their kid through a normal day in school, the bulkiness of Braille materials, the stresses on the family, and the loss of productive members of society. And so the RAC voted unanimously to approve us. And the net result is a good one, not only for us, but the other trials that initiated gene therapy then began to enroll children as well. So we also are lucky to have a great IRB, and this IRB gave us approval based on this particular code, this legal code. And they also did us a real favor because we had proposed to start at a dose that was probably lower than we would like to achieve a therapeutic effect. And the IRB pointed out to us that because more than 90% of our dogs showed benefit at this particular dose, our low dose study, that we should start on this dose with children, that optimizes the risk-benefit issue. And so we did. And so I think that's one reason why we began to see efficacy even at the first dose. This trial was a dose escalation study with 3 doses, each dose differing by half a log unit. And it's, of course, involved individuals with RP65 mutations, children and older, and their current follow-up is now more than 6.5 years. At one point I'd like to make is that our collaborators were key to identifying the initial set of subjects. People were at first hesitant, I think, in this country to enroll in a gene therapy trial, but thanks to our European collaborators, we had no trouble enrolling quickly, and then the United States citizens followed suit. So it's been an excellent safety profile, and fortunately there are no severe adverse events due to vector, and fortunately we haven't had any severe adverse events due to the change in lifestyle of our patients. You can see here, which is somewhat horrifying to me, but you can see they've gone on with gusto, enjoying life, although they tell me now that when they do things like this, skydiving, they do it with their eyes closed. So one question is where to inject the macula in the periphery, and Dr. Drack is going to address this in a few minutes, but this has obvious implications in terms of the outcome, in terms of the roles of different portions of the retina. But we decided, or the surgeon, Dr. Albert McGuire, decided that the best possible outcome would be the macula, and so he started the patients injecting the macula, and our second patient had an asymptomatic macular hole that was found 2 weeks after injection, not immediately upon surgery. He didn't know it was there. He actually came in after reading hand motion, having hand motion vision, came in reading 6 lines on the eye chart. And so obviously he was happy with his results, but it wasn't something that we wanted to have happen. And so Al and colleagues revised the surgical procedure to select an injection site more distal from the fovea and to take steps to remove membranes which might cause stress on the retina. And so far we have not had similar complications. And this is an issue that Robert McLaren addressed really beautifully a couple days ago as well. But one of the things that we think is important is to share complications. If you're a doctor, if you're a surgeon, you'd be foolish to say you're never going to have any complications. And I think it's the complications that we can probably all learn the most about. And so we went ahead and told other clinical trials about this complication, warned them about it, and actually have a cover page showing our complication with the macular hole right front and center. So what are the results of our study? This graph on the top shows the visual function with age in terms of visual acuity, reading the eye chart, and the one below it shows the changes in light sensitivity. And the subjects are arrayed from youngest to oldest in both of these graphs. And in the visual acuity testing, you can see that more than half of the subjects showed a statistically significant improvement in visual acuity. That's reading more than 3 lines on the eye chart. And that all of the subjects showed a significant improvement in light sensitivity, with the youngest individuals showing the biggest improvement. And I'd just like to point out that that person I showed you who had the macular hole showed improvements in both visual acuity and light sensitivity. So that, again, did not alter his benefit. We also used objective data, shown here is the pupillary light reflex, which is a reflex, and so the person isn't telling you whether or not they're responding. And this is really robust and gives quantitative information in terms of amplitudes of response and also comparison of the 2 eyes. And you can see in this person who was injected in the right eye, the left eye is illuminated, no response. Right eye is illuminated, brisk response. And there's a brisk response every time the right eye is illuminated because that eye is now responding to visual cues. And this is a finding that we found that was improved in all of the subjects who received the injection. We also developed a mobility course to look at functional vision because we didn't want to send people across a busy street and see if they could navigate better or down a stairwell and get hurt. We needed something where they could, we could set this up in the confines of a exam room. And so shown here is this setup where the person starts at the start point, follows arrows, takes turns, goes up steps and down steps and avoids bumping into different obstacles. And the course is rearrayed for every single test so that the subjects don't memorize the course. And just to show you some of the results, these show the results for actually the first child in the gene therapy study, where he is going through at baseline, bumping into every object there is. And if you read the text below, this is when he spoke Dutch. Before he spoke English, he says he doesn't see anything. And then in comparison, he goes through the course after the treatment, let me see if I can get this to go. You can, well, if I could get that to play, you would see that he walks through this course without any difficulty at all. So you'll just have to believe me on that one. We also looked at plasticity of the visual pathway. And this is done in collaboration with Manzar Ashtari. This is a study that was done as ancillary to our clinical trial studies, looking at the effects of treatment on visual cortex activation. And there we could take advantage of the known retinotopic pathways between the retina and the visual cortex and stimulate the right, in this case, untreated eye, where we don't see any activation after gene therapy, whereas after the left eye is illuminated, you can see there's robust activation, which maps out specifically to the area of the retina that was injected. And this has also been a phenomenon that we've seen in multiple patients and confirms the fact that these people are seeing, but it also it gives us a window into the incredible plasticity of the central nervous system. And one of the challenges with doing these ancillary studies, in particular fMRI, is the challenges of incidentalomas, findings that you are not looking for that may be apparent in the imaging. And this, of course, has been annoying, and because it's a clinical trial, requires elaborate reporting. And so it's something I just want to point out to people who are planning clinical trials is if you are doing all these ancillary studies, remember that everything has to go onto a form and be reportable. So what about safety and efficacy post-readministration? We thought, well, if we see this benefit in one eye, wouldn't it be better if we actually treated both eyes and get more improvement in both eyes? So this was a big question to us, and one that really made us nervous because there's been very limited success in readministering gene therapy in other organ systems. Of course, the concern is a toxic immune response, because if the gene therapy reagent is delivered once, the body could develop an immune response to it, and then if you come back a second time, this could cause toxicity not only to the newly treated eye, but it also could limit the benefit in the initially treated eye. So we went back to the laboratory and carried out numerous large animal studies, first on affected dogs and then on non-affected monkeys. And the studies were designed such that one eye was treated with a very high dose. We'd come back 2 or 3 weeks later when we'd expect immune response to be maximal, treat the second eye, and so forth. And as you can see here, this is one of our subjects who did quite well even after both eyes were received the intervention. And this dog is actually living happily in our home right now. So the results of the phase 1-2 studies that were the readministration studies are shown here. As I mentioned earlier, people have been followed for more than 6.5 years and more than 3 years since the second eye was treated. And the safety profile has been benign. The adverse events have been minimal, and there are no severe adverse events deemed related to the vector itself. In fact, some of the individuals who at first were unable to navigate the obstacle course after the first eye had been injected were able to after the second eye was injected. And you just saw the post-readministration movie here, and this is the before administration movie where the subject immediately went off course. So where are we now? We decided to move forward to a phase 3 clinical trial, and that's the phase where it's a pivotal study where in this country, the FDA determines whether this product can be prescribed by physicians for eligible individuals. And it actually took us 2 years to figure out how to design this phase 3 study. And we went through very helpful conversations with the FDA in designing it. What we ended up doing was designing an open-label, randomized, multi-center controlled trial, which enrolls more than 24 subjects, ages 3 and older. The FDA was concerned that subjects with this particular condition might improve on their own. And so we built in a control group shown here. People are randomized 2-to-1 either to intervention or control. And then the control group is followed for a year with the same follow-up testing as the intervention group. And then at the 1-year point, they switch over to the intervention group and get both eyes injected as the intervention group would have. And again, go through follow-up testing, and then they're followed up through year 15 as mandated for gene therapy studies. We knew that if we had a separate control group with no chance of intervention, that nobody would sign up for that group, since it was pretty well known about the results of our clinical trial. And the other issue about this trial is that both eyes are injected. We initially had proposed injecting only one eye so we could use the contralateral eye as a control for an internal control. But the FDA argued, probably reasonably, that most retinal surgeons, when this is prescribed, will want to inject both eyes. And so that is the paradigm. And this trial is well underway, and we expect to have the first set of results in 2015. The data is analyzed by, the primary outcome measures is analyzed by outside analysts who are masked to whether people are in the intervention group or the control group. So I can't give you this data for fear of In fact, I don't even know all of the data just to keep all of the results clean and the analyses clean. So that sums up what I wanted to tell you about. And now we're going to move on to Dr. Catherine High, who is at Children's Hospital of Philadelphia and has been involved in gene therapy for more than 25 years, amazingly enough. Her initial expertise has been in hemophilia, and she is one of the world-renowned experts in gene therapy. Thank you, Kathy.